not a week from today. But if today were Friday, it would be a week from today. All right, so we've got a fair amount of material to cover. We're a little bit behind, but not a problem. Last time I got started talking about the signaling through the beta-adrenergic receptor, which is shown right here. And we saw how that a signal that comes from outside the cell can cause some big changes inside the cell. And I talked about how protein kinase A becomes activated. I haven't yet talked about what protein kinase A does. All right? So that will come later. But suffice it to say that protein kinase A's action, as far as this class is concerned, will be activating some enzymes that are involved in breaking down glycogen, which is how the cell is making, or the body is making glucose available in the bloodstream. So we'll talk more about that when I talk about glycogen metabolism, where all of this stuff will hopefully start coming together. Okay. There's a G protein. Memorize that structure and write it for the exam. The G protein has 3 subunits, as I mentioned last time. It has an alpha subunit, it has a beta subunit, and a gamma subunit. And the beta and gamma bind to the alpha, as you see shown here schematically. And they, in doing so, cover up a binding site for the adenylate cyclase. And so it's the combination of the loading of the GTP and the release of the beta and gamma subunits that allow the alpha subunit bound to GTP to connect to the adenylate cyclase and favor the synthesis of cyclic AMP. Okay? As I mentioned last time, the G proteins are bad enzymes, and they're bad enzymes bad in the sense that they're inefficient, and their inefficiency helps them to turn themselves off. And as we'll see later today, the inability to turn themselves off at times can cause some major problems. So that's important to keep in mind. All right. The overall epinephrine signaling pathway looks like this in words compared to what you saw in the schematic diagram. So I don't care how you learn that. One way of learning it, I find the figure is a little more helpful. But you notice that there's several levels of amplification, and these amplifications happen through the synthesis of molecules or the activation of enzymes, so that at each level we get an increase in the amount of signal. And this increase in the amount of signal is what we saw in the cascade, for example, for the blood clotting proteins. And that signaling cascade allows a very small signal to be amplified very much and very rapidly. And so this is why adrenaline acts fairly quickly. The major limitation in the action of adrenaline is not the action that occurs inside the cell, it's actually the getting it to the cell, because adrenaline has to travel through the bloodstream. Okay. Resetting G proteins is important, and that's related to their, what I described as their inefficient enzymatic action. You can see here GTP bound to adenylate cyclase. Over time, that enzyme will, in fact, cleave that GTP to create an alpha subunit that has GDP bound to it. That GDP subunit will be less, GDP bound subunit will be less inclined to bind to adenylate cyclase and will later link up back up with the beta and gamma and go back to the original state that we talked about before, where it'll be adjacent to the beta-adrenergic receptor. Okay. Now, signal termination is pretty important, right? So you saw last time I talked about the importance of phosphodiesterase, because phosphodiesterase is present in cells at a low level. And so over time, it breaks down cyclic AMP. So if cyclic AMP gets built large amounts, over time the phosphodiesterase will take over and start breaking that down. Well, that's only going to work if the adenylate cyclase stops making the cyclic AMP. And that means that the very beginnings of this signal also have to have some control. So cells have a sort of a backup mechanism of ensuring that the binding of the hormone to the receptor on the cell surface originally, making sure that that binding actually does get turned off. And so this is what I'm showing you on the screen is a sort of a backup mechanism of turning off that signal that's coming from the beta-adrenergic receptor. you can see in the scheme of things that a receptor lacking a epinephrine can bind to one and get put into an activated state. That's you've already seen before. You can also see that the release of that epinephrine can occur, as you see here, by the process described as dissociation. But if dissociation doesn't happen well, let's say this hormone gets stuck in here, and this receptor got stuck in the on state, that would be problematic for the cell because it would be continually stimulating the cell to make cyclic AMP, to activate protein kinase, to continue to produce glucose, and that would cause the cell ultimately to die because it couldn't do that forever. So one of the ways of ensuring that that doesn't get stuck in that on mode is the process that you see on the screen. And this is through the action of something called a receptor kinase. A receptor kinase can recognize that this beta-adrenergic receptor is stuck in the on state. And if it detects that, what it will do is it will phosphorylate a couple of sites on the inside of the cell. Remember, this is the outside above and the inside below. It'll phosphorylate a couple of sites on that beta-adrenergic receptor, putting phosphates onto some amino acids in the adrenergic receptor. And those phosphorylated sites become the target for this protein called beta-arrestin. And when beta-arrestin binds to this complex, it basically stops it from interacting with G proteins so that that signal is stopped, dead in its tracks. So beta-arrestin has a very simple name. It's arresting the process, and it's very easy to recognize for that purpose. Okay, so this is yet another way of turning off that signal. So you can see that cells really place a lot of importance on turning on and turning off the signals. Another mechanism that is a signaling mechanism that's found inside of cells involves activation of another enzyme. And this also works through a 7-TM. I don't have a figure showing you this 7-TM. But this 7-TM ultimately activates an enzyme called phospholipase C. Now I'm talking about a different signaling mechanism here. This signaling mechanism may play roles in constriction of blood vessels, for example, and there's a variety of ways in which it can act. But the bottom line of this signaling pathway is that the activation of this pathway activates an enzyme on the inside of the cell called phospholipase C. Phospholipase C catalyzes the reaction that you see on the screen. And this reaction that you see on the screen is interesting and important. Okay? So if we look at the substrate for phospholipase C, it's this molecule up here, phosphatidyl inositol 4,5-bisphosphate, blah, blah, blah. You can call it PIP2. PIP2 is a very common molecule that we find inside of cell membranes. Very common molecule found inside of cell membranes. So this is very abundant inside of cells. And when phospholipase C is activated, it breaks the bond between this compound up here shown in red, which is IP3, to give it a short name, and the remaining portion, which is diacylglycerol, or DAG. All right? Now, it's important here because both of these molecules act in a signaling capacity. They both act as second messengers. So this signaling pathway that I'm describing to you here involves creation of 2 second messengers, and it happens through the activation of phospholipase C. Now, what happens in this process is that you see the breaking of the bond between these two things here. The IP3 is, as you can see by its chemical structure, very polar, it's very water-soluble, and it's very much able to travel in the cytoplasm. Remember, this guy is found in the membrane. So this whole complex up here is starting in the membrane, cleaving of the compound by phospholipase C, releases IP3, which now can travel in the cytoplasm. Okay? What's left behind in the membrane is this molecule called diacylglycerol, or DAG. As I said, both of these compounds will act in this mechanism I'm going to describe to you as second messengers. Okay? So one left in the membrane, one travels in the cytosol. All right? This depicts schematically what I just described to you. All right? Here's the membrane. Here's the PIP2 that you can see in the membrane with the attached inositol compounds. Okay? Activation of phospholipase C causes this IP3 to be released into the cytoplasm. Okay? The release of IP3 into the cytoplasm causes IP3 to freely go to the endoplasmic reticulum. And at the endoplasmic reticulum, there is a receptor that binds to IP3. So we see a receptor, and now IP3 is causing endoplasmic reticulum to open up its gates. And when it opens up its gates, one of the things that's stored in the endoplasmic reticulum is calcium. What's happening here is this signaling mechanism is causing the calcium concentration inside the cytoplasm, causing the calcium concentration inside the cytoplasm to increase. I've told you previously that calcium was kind of a critical ion inside of cells. Cells manage the levels of calcium because too much calcium causes DNA to precipitate. So that means that the calcium concentration inside the cytoplasm starts out pretty low. The cells are pumping calcium into the endoplasmic reticulum to keep the cytoplasmic concentration low. So when this signaling process comes along, it opens up the endoplasmic reticulum, calcium concentrations rise. Calcium concentrations rise, the calcium can bind to another kinase called Protein Kinase C. You're getting tired of protein kinases here. Protein Kinase C is located at the membrane, and it's bound to the other second messenger, diacylglycerol. We can kind of think of calcium here as a third messenger because it was released upon the release of the second messenger, which is IP3. Okay? What is happening here is the binding of protein kinase C by both diacylglycerol and calcium causes protein kinase C to become activated. And activation of protein kinase C can cause many things to happen in the cell, including gene expression, specific gene expression. So here's another signaling mechanism. This signaling mechanism isn't quite as targeted as the one I described to you before for the beta-adrenergic receptor, but we can see activation of a protein kinase, and that protein kinase can cause many things to happen in the cell. Again, a lot of things happening as a result of binding of a hormone to the cell's surface. Okay. So signaling mechanisms, as you can see, are multi-step. They involve messengers, and those messengers travel through the cell in order to make things happen. A very common occurrence of signaling is activation of a kinase. And we don't go through them in this class. I'll show you an example later, but we'll see that kinases can activate kinases, can activate kinases, can activate kinases. That is, we can see a whole series of these, again, not unlike that we saw with the protease cleavage that happens in blood clotting. Okay? That's amplification of signals, a very important component of the signaling process. Okay, well, calcium is important, and calcium is also something that I've told you that the cell has to be careful with. The cell has to be careful with calcium because, as I said, too much calcium, chromosomes precipitate. Right? So when calcium is released in cells, cells actually have proteins that will gobble up that calcium and stop the free calcium concentration from getting too high. One of the proteins that's very important in grabbing a hold of calcium is a protein that you see on the screen, schematically shown as what's called an EGF hand protein, but the specific protein is called calmodulin. Calmodulin. Calmodulin is a small peptide that has a structure kind of like this. These are called EF hands. And right here is where the calcium will bind. Now, the binding of calcium by a calcium binding protein, not surprisingly, causes the calcium binding protein to change shape. Causes the calcium binding protein to change shape. Here's calmodulin without the calcium. Okay? We see the binding of the calcium that causes a somewhat of a structural change, and that small structural change that's happening here on the binding of calcium facilitates a bigger structural change that allows the calmodulin to bind to another protein and activate it. Okay? So I'm going to step you through this. We've gone through the phospholipase C pathway. We've released calcium in the cell. The calcium has bound to a protein called calmodulin, caused a small structural change in calmodulin. That structural change was enhanced by another enzyme, and that binding of the other enzyme by calmodulin activated it. The consequence here is that calcium is activating another protein. Just like cyclic AMP was activating protein kinase A, calcium is activating indirectly through the binding of calmodulin another enzyme. Now the beauty here is, again, a protein is binding calcium. That leaves the free calcium concentration relatively low, yet still a signal is being propagated as a result of the calmodulin's change in shape that's happening. Questions on that? Yes, over here. Is the calmodulin activated or the protein activated? So when we talk about activation, it's all a relative thing. Calmodulin is changed. You can see the gaining of the shape that's happening here in the middle compared to what it was. So calmodulin has changed its shape. Calmodulin doesn't catalyze a reaction, though. So I don't think of calmodulin in terms of activation. All right? But calmodulin has definitely changed from the state that it was in, and it's activating this other enzyme as a result of that shape change. Make sense? Yes, Garrett? Is there a reverse process to this? What do you suppose? Okay. How would we reverse this process? Good question. Yeah. So as we start removing the calcium, remember these are not covalent bindings, so when we have non-covalent bindings, these things can come back out. This guy will lose its calcium, and when it loses its calcium, it's going to go back to this state here. It's no longer going to activate that enzyme, and we reverse the process. Now, cells have, as you will see next term, various what I call pumping mechanisms for taking calcium and pumping it back into the endoplasmic reticulum. That's important because it had to get there in the first place somehow, right? So those pumping mechanisms will take over in time, take the calcium, pump it back into the endoplasmic reticulum, thereby reducing the calcium concentration in the cytoplasm, and thereby reversing this process. Okay. What you see? Yeah, question. APO means no ion bound. APO means empty, literally. Yeah. Okay. Well, let's talk about perhaps a bit more interesting kind of signaling process that happens. The signaling process I'm going to talk about now relates to insulin. Okay? And insulin signaling does not occur through a 7-TM. The first two I've described to you occurring through a 7-TM. This process does not occur through a 7-TM. It occurs through something called the insulin receptor, and the insulin, that's insulin right there. The insulin receptor looks schematically like this. Okay? It's not a 7-TM. It is a transmembrane protein, as all receptors are. And if we look at this, we see an outside the cell on the top. We see an inside the cell on the bottom. Insulin is an example of a receptor that's what we call a tyrosine kinase mediated receptor. Tyrosine kinase mediated receptor. Now, the kinases I've talked about so far, remember a kinase means put phosphate onto. The kinases I've talked about so far put phosphates onto serines and threonines. They don't put phosphates onto tyrosines. But I told you when I first talked about kinases that some kinases did put phosphates onto tyrosines. This is the first kinase that we're seeing that puts phosphates onto tyrosines. Okay? Now, I'm going to step you through what happens with this process. And what you're seeing in these signaling pathways is this goes to this, goes to this, goes to this, goes to this. Okay? That's what happens in signaling. All right? I do spare you some of the details, but wherever I talk about this going to this, going to this, going to this, then yes, you should know that. Okay? Okay. What happens in insulin signaling? Well, first of all, let's think about what's happening with insulin in the first place. Remember I said last time that glucose is a poison. Glucose is a poison. Your body treats it as if it's a poison, and that's because it acts as if it's a poison. High levels of glucose, which happen in diabetes, have very detrimental effects for people. Can lead to blindness, especially among the elderly. It's not uncommon for the high glucose levels to result in amputation of limbs. And these high levels of glucose are the cause of the problem. Okay? Glucose is a poison. In your bodies, if you're not diabetic, what's happening is insulin is helping to modulate the levels of glucose that you have in your bloodstream. You go eat a meal, your blood glucose levels start rising, your body says, poison. It starts making insulin, and insulin causes cells to take up glucose and reduce the blood glucose levels. And you might say, that's an odd kind of poison. It's a poison that the cell is taking up. Isn't the cell going to die? And the cell is not going to die, obviously, because it takes that insulin and it does something to it. It modifies, I'm sorry, not the insulin, it takes that glucose and it does something to it. It modifies that glucose so that it's no longer glucose. Again, consistent with it being a poison. The cell is literally detoxifying that glucose within inside of itself. Okay? Now, let's look at the signaling process that makes this all happen. Eat a meal. Drink a Coke. Get one of those God-forbidden lattes that you get. All right? Blood glucose levels go boing. Pancreas says, okay, got to release insulin. It releases insulin. Insulin travels in the bloodstream and goes and hits target cells that have a receptor, binds to them, and then causes a series of events to happen. Okay? You'll notice that the insulin receptor is what we call a dimer, meaning it has two identical sets of proteins, and each protein is multi-subunit. There's the beta subunits in yellow, there's the alpha subunits in blue, and these are shown as a dimer. They exist in the cell membrane as a dimer. Okay? I told you that this receptor is a tyrosine kinase. And to remind you what a tyrosine kinase does is it takes the side chains of tyrosines and puts phosphates on them using the ATP from, using the phosphate from ATP. Okay? Now, yep, that's not what I want. Here we go. This shows the overall process I'm going to tell you about. Okay? Here is an insulin receptor that is bound to insulin. The binding of the insulin by the receptor causes one subunit of the receptor to put a phosphate onto the tyrosines of the other subunit. This only happens when the insulin binds to the receptor. If there's no insulin in the receptor, the first subunit does not put phosphates on the other one. They sit there sort of intertwined, blocking each other's active site. But the binding of insulin caused them to change shape so that one of them has its active site open to phosphorylate the other one. Doesn't matter which one for our purposes. Okay? When one of these subunits puts phosphates on the tyrosines of the other one changes shape and starts putting phosphates back on the first one. So they're phosphorylating each other. You can see there's multiple phosphates that get put onto there. And those phosphates become the target for binding of a protein called IRS1. Sounds like it's a tax thing, doesn't it? Okay. IRS1 recognizes this phosphorylated tyrosine. Okay. It recognizes this phosphorylated tyrosine and binds to it. Okay. This binding of the phosphate by the IRS1 causes the tail of IRS1 to be brought into close proximity to the kinase activity in the beta subunit. That's how these phosphates get put onto there. Okay? That's how these phosphates get put onto there. So these are also phosphorylated tyrosines. These phosphorylated tyrosines are targets for binding of this guy here called phosphoinositide 3 kinase. And phosphoinositide 3 kinase is a membrane-associated protein that is activated by two things. One, a phosphotyrosine here on IRS1, and second, PIP2 in the membrane. It takes PIP2 and it cleaves it. I'm sorry, it doesn't cleave it. It phosphorylates it. I got it backwards. It phosphorylates it, okay, and makes PIP3. That 3 means an additional phosphate's been put on to that molecule. PIP3, in turn, is a target for another protein kinase. You guys love signaling, I can tell. PDK1. PDK1, in turn, is a protein kinase which now activates another kinase, another kinase, another kinase. And ultimately, what this signaling pathway does is it changes the movement of a very critical protein inside of the cell. The movement. Okay? We haven't talked about protein movement other than in general terms so far. But proteins can travel in the cell. You may remember I said they had license plates that we could tell where they were supposed to go. This kinase cascade that's here changes the movement of a very important protein called GLUT, G-L-U-T. There are several GLUTs in the cell. GLUT stands for glucose transport. What this kinase pathway is doing is it's altering the movement of GLUT in the cell. Prior to this insulin binding, GLUT's floating around down in here. After the insulin binding, this whole pathway kicks in and GLUT gets moved to the membrane of the cell. And GLUT, you recall, has a name, glucose transport. What's happening is we've now told this cell, bring in glucose. The glucose transporter in the membrane starts seeing all this glucose that's out there and starts bringing it in. And what happens to the blood glucose levels? They fall. All right, I'm going to step you through it again, so don't frown too much, okay? Let's start with the bottom line. The bottom line is release of glucose. Back up. Release of insulin stimulates a process that results in the movement of a glucose transport protein to the cell membrane. That's the big picture. Because that glucose transport protein is in the membrane, the cell takes up glucose. And if we do this on an organism-wide action, then glucose levels will fall because the cells are taking up glucose. All right, everybody see the big picture? The rest of this is details. Okay? The details are binding of insulin activates the tyrosine kinase of one of the units, which phosphorylates the other, which phosphorylates it back. That makes these units completely active. They, in turn, phosphorylate IRS1, which has recognized a structure on them and bound to them. The phosphorylated tyrosine on IRS1 is a target for phosphoinositide 3 kinase. Phosphoinositide 3 kinase is activated, makes PIP3. PIP3 is bound by PDK1. PDK1 stimulates the kinase cascade. Yes? Question about whether diabetes is a result of a problem with the insulin receptor or insulin itself. The problem with diabetes is actually quite complicated. There's different kinds of diabetes. Type 1 diabetes involves inability to make insulin. And if you could imagine, the inability to make insulin means blood glucose levels are going to go, whoa, and you're putting a lot of poison in the bloodstream, causes a real problem. Type 1 diabetes is not the most common type of diabetes. Type 2 diabetes is much more common, and type 2 diabetes manifests itself in several ways. People with type 2 diabetes make insulin, but their body doesn't respond to it appropriately. They become what we call insulin resistant. So even though the body is making insulin, this process that I've described to you here doesn't occur as efficiently until a ton of insulin is made. Okay? And so that means blood glucose levels will go very high if your body's not responding, and then all of a sudden, when you get to a high enough level, the body starts taking up glucose, and what happens is it goes very low. So people who have type 2 diabetes may have very high blood glucose levels and very low blood glucose levels, hyperglycemia and hypoglycemia. Some people with hypoglycemia, type 2 diabetes, may have to take around a sugary drink or something with them in case their blood glucose levels fall too low, because they can faint and lose consciousness as a result of that. Does that answer your question? Yeah, question.
Obviously, type 1, you can treat the shot. How do you treat type 2?
Okay, so how do you treat type 2? There's no one answer to that question because there's no one type 2 diabetes. All right? Insulin is one way of dealing with that because, again, levels of insulin do have factors here, and you can boost that by taking that. More commonly, you try to manage it with diet. So you've really got to be careful with diet, and that's a very big consideration. Yes? PIP2 is acting with IRS. That you're right as well. I didn't mention that. Yes, sir?
So what happens, can IRS1 bind to multiple phosphates on the
Say it again.
Can IRS1 bind to multiple phosphates?
Can it bind to multiple phosphates here?
Multiple IRS1 binding.
You can have multiple as long as you've got the appropriate phosphates exposed. They can bind there, yes. Mm-hmm. Up top, yeah. Okay, so the question is the uptake of glucose passive transport or active transport? It depends on the type of cell, actually. Blood cells have gluts, for example, that can allow passive transport to occur, and other cells may require active transport. So it's not one straight answer. Question somewhere? Yes, Stephanie. Yeah, our question, it's actually a good question. I sort of describe this glut as if it's floating around the cytoplasm, and that's not really accurate. The glut protein is contained typically within the endoplasmic reticulum, and so we're talking about a movement. You notice how I showed the other day the movement of things from the endoplasmic reticulum to Golgi apparatus to the membrane, for example. That will facilitate that movement much more readily. So it's not fundamentally changing the hydrophobicity or the hydrophilicity of the protein, which was what her question was. Good question, though. Okay, so insulin signaling is interesting and important. One term I didn't give you, and I should give you, I want to make sure, is that this domain of a protein that recognizes phosphotyrosines has a name. It's called an SR2 domain. SR2 domain. That's a specific structure that's found in many proteins, and that specific structure recognizes phosphotyrosine. Phosphotyrosine, as we shall see, is very important in many signaling mechanisms that affect cells' choices to divide or not to divide. What I'm showing you here with the insulin receptor, I should point out, is only one of a zillion things that the insulin receptor can affect. Insulin receptors are implicated in processes that include cancer, cell division, and a variety of things. So insulin receptor is a very, very important and multifunctional receptor. Insulin, in many ways, is there because glucose is a poison. And insulin itself, the more we produce insulin, may be also indicative of other problems. So we want to, if we think about things relative to high-carb diets, low-carb diets, okay, maintaining low-carb diets allows us to keep our insulin levels at a relatively low level, and there probably are some very good reasons to do that as well. So there is some truth to the fact that maintaining low-carb levels probably are good. Getting crazy with that, no, but monitoring your carbohydrate levels is good. Here's the SH2 domain that I mentioned briefly, and it is a specific structure in a protein that recognizes phosphotyrosine, as I said before. You can see there are some arginines that are involved in that recognition, and they interact with the negatively charged phosphate on the phosphotyrosine. Okay. We'll pass on that. There's the words of what I just told you. Okay. The next signaling mechanism I want to talk about relates more directly to cell division. And this one, you'll start to see, again, how these kinases play roles in the overall regulation of a cell. Okay? The system I want to talk about here is called the epidermal growth factor system. Okay? So the epidermal growth factor is a hormone. It is a hormone that can stimulate cells to divide. It can stimulate cells to divide. And it works by binding to a receptor in the membrane of cells. You've heard this before. And the receptor that it binds to has the very confusing name of epidermal growth factor receptor. Okay, that's a joke. All right? So epidermal growth factor binds to epidermal growth factor receptor. The receptor is shown here. And if you look at that, you say, boy, that sure kind of looks like that insulin receptor. And from a schematic point of view, it does. There is a big difference, though. One of the big differences with the epidermal growth factor receptor is it does not normally exist as a dimer in the membrane. It exists as a monomer. You see it as a dimer here because this receptor has bound to epidermal growth factor. And when one epidermal growth factor binds to when one receptor for epidermal growth factor binds to epinephrine, God, I can't say it. When one EGF receptor binds to EGF, its shape changes slightly. The shape change involves this little red thing right here. The red thing is normally folded up inside the cell. But when an EGF receptor binds to EGF, this red thing sort of sticks its hand out. Well, it takes another receptor binding to EGF to stick its hand out, and the two hands interlock. That's how the dimer forms. If there's no EGF bound to the EGF receptor, the hands are closed inside themselves, and these receptors just float through the membrane as monomers. So it's the binding of the EGF by the EGF receptor that causes the monomers to extend their hands and dimerize. Everybody clear on that? The binding of the receptor causes the dimerization. That's different from insulin. Insulin, the receptor was already a dimer. Well, you're going to like this mechanism because the dimerization of this receptor causes a series of events to happen very much like what happened with the insulin receptor. The EGF receptor is also a tyrosine kinase. The tyrosine kinase phosphorylates each other, causes each one to become more active. The phosphorylated tyrosine of the receptor is a target for this protein called GERB2. GERB2 has an SR2 domain, recognizes phosphotyrosine, binds to it. Another protein called SOS binds to GERB2 only after it has bound to phosphotyrosine. So the series of events, EGF receptor dimerizes as a result of each unit binding EGF. The dimerized receptor causes tyrosine protein kinase to become active, so the tyrosines on each receptor become phosphorylated. Those phosphotyrosines are targets for binding by GERB2, which has an SR2 domain. GERB2 bound to this is a target for binding by SOS. Help! And now we get to the meat of this. SOS is target for binding a really interesting protein called RAAS. RAAS, R-A-S. Now, RAAS turns out to be a fascinating protein. Our cells have several different forms of RAAS, but each of those forms of RAAS are involved in activating or helping the cell to make the decision, do I divide or not divide? Okay? So this process here is important. If this signal makes it all the way through, RAAS becomes activated, and I'll explain that in a minute. And the activated form of RAAS is a signal to the cell, they're telling us to divide. Let's divide. Okay, so we can imagine you're a growing individual. You need to make new bone cells, for example. Your body says, hey, we need to grow. It starts releasing growth hormone. Growth hormone comes, activates the process. Bone cells start dividing, and you start growing. Okay? That's if everything's working fine and dandy. Okay. We can imagine that we can screw this process up. If we screw this process up and we activate RAAS, when we really don't want to activate RAAS, we're going to have growth that's out of control. And if that signal is always on, then we're going to have a cancer. Okay? So the first thing I'll tell you about RAAS is something that we call a proto-oncogene. Proto-oncogene. A proto-oncogene has a very nasty sounding name, but it turns out to be a pretty innocuous thing. A proto-oncogene is a gene whose protein product, in this case RAAS, whose protein product plays some central role in an important cell decision. This case being growth, and commonly growth and division are the important roles that proto-oncogenes play. Your body is full of proto-oncogenes because your body has to control cell division. All right? Now, I'm going to tell you how one way we can screw that process up. Well, let's talk about what's happening here in this activation first. You might look at this and say, boy, that sure looks like a G protein, Kevin. Because G proteins were things that bound to guanine nucleotides, and when they got stimulated, they replaced a GDP with a GTP, and they became active, and that activation caused something to happen, and the answer is that RAAS is a type of G protein. RAAS is a type of G protein. It binds to guanine nucleotides. It gets activated by the binding of GTP. And just like G proteins, RAAS is an inefficient enzyme. What does it do? Well, like the G proteins, it cleaves GTP back to GDP. So this signal turns itself off unless something happens. Okay? If something stops this enzyme from converting GTP back to GDP, this RAAS is going to be left in the on signal, and the cell is going to be continually getting the message, divide, 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 divide. How does that signal get stuck on? A really dandy way is mutation. Okay? Mutation, change in the sequence of DNA, can change the sequence of amino acids in the protein that's coded by that DNA. If we mutate a proto-oncogene, we can change the proto-oncogene's function so that it no longer is functioning right, and now the cell divides uncontrollably. We have just created an oncogene. Get rid of the proto. Oncogenes are the result of mutation. Proto-oncogenes are the starting things, unmutated. I'll give you a little scary thought before we go to our song for the day. Okay? The mutation that can cause RAAS to not be able to cleave GTP involves the change of a single base inside of the RAAS coding. Amino acid number 11, I believe it is, normally has a glycine. If you mutate that glycine to any other amino acid, this protein will leave RAAS in the GTP state. That's one mutation, folks. When you wonder why people worry about pollution and the air they breathe, the food they eat, the water they drink, the reason is because they don't want to mutate any more than necessary, because one mutation, you're hosed. Not a pleasant thought. That's not a pleasant thought. Quick question. Shh. Did I say SR2? I said SR2. I meant SH2. I apologize. Yeah, the gene it's named for is SARC, which is SR, and I was thinking that when I said that, but it is SH2 domain. Okay, this is a song about signaling. Oh, Limerick. Oh, my God, I forgot. I'll do 2 next time. This one's to the tune of The Sound of Silence by Simon and Garfunkel. Biochemistry, my friend, it's time to study you again. Mechanisms that I need to know are the things that really stress me so. Get these pathways planted firmly in your head, Ahern said. Let's start with epinephrine. Membrane proteins are well known, changed on binding this hormone, rearranging cells without protest, stimulating a G-alpha S to go open up and displace its GDP with GTP because of epinephrine. Active G then moves away, stimulating add cyclase. So a bunch of cyclic AMP binds to kinase and then sets it free. All the active sites of the kinases await triphosphate because of epinephrine. I can't carry a tune. Muscles are affected then, breaking down their glycogen. So they get a wad of energy in the form of lots of G1P. And the synthases that could make a glucose chain all refrain because of epinephrine. Now I've reached the pathway end, going from adrenaline. Here's a trick I learned to get it right, linking memory to flight or fright. So the mechanism that's the source of anxious fears reappears when I make epinephrine. Okay, guys, bad singing.